So today's lecture is on the cell membrane and transport. We're going to talk about the components of the cell membrane, characteristics of those components. We're also going to talk about how things cross the membrane and why, based on their characteristics. Is it easy? Is it difficult? And lastly, we're going to finish with things that are that massively cross the membrane in mass quantities, both going into the cell and out of the cell. So the first thing we have to do is we have to talk about what the main structure of the membrane is made of. We've talked about this already before when we talked about lipids, but phospholipids are important components of the cell membrane. They are amphipathic molecules, meaning that they have hydrophilic parts and hydrophobic parts. Those hydrophobic parts are the tails, which contain long chains of carbon and hydrogen. And then you have the hydrophilic parts, which are the heads, which are much more polar and even sometimes have a charge associated with them. And as a result, water has an affinity to them. So if you look at a bilayer, right, the lipid bilayer, which is called that because you have the hydrophilic heads pointing towards the watery environment, both outside and inside the cell, and the tails pointing to each other, you can see we have kind of two layers here. And if you look at proteins that are embedded in the membrane, which is another component of the cell membrane, because proteins are, as we're going to talk about, have tons of different jobs, the hydrophobic region of the protein is going to have many R groups within it that are long chains or are made up of just carbon and hydrogen, because that's what makes something hydrophobic, because of that equal charge distribution. On the top and on the bottom, on the parts that are towards the heads and pointing towards the watering environment, you're going to find R groups that are much more hydrophilic, right? So you're going to see R groups that have oxygens and other elements in them that are going to make them draw electron density unequally through the R group, and as a result will fold and orient themselves towards that watery environment. So the cell membrane is the outer covering of the cell in general. There's also a nuclear membrane that's made of phospholipids, and most of the organelles we're going to talk about, there's some debate whether even certain things that don't have membranes are considered true organelles, but an organelle is something that is pretty much covered in this lipid bilayer. We're going to focus, as I said, on the plasma membrane or the cell membrane, the outside of the cell, but all of these kind of have and exhibit this selective permeability, which is that certain things are let in and certain things are let out. And what really dictates that is the type of molecule itself. Is it polar, nonpolar, charged? Is it large or small? And what type of proteins are in the membrane that act as channels or kind of doors or vaults that let certain things in or out. So selective permeability is that certain things are let in or out. It's also, the membrane exhibits what's called fluidity. Fluidity is just flow and movement. And phospholipids can slosh and move around constantly within the bilayer. You can see kind of in the GIF that I presented there, though it's not super long, you can see it has tons of proteins embedded, but that bilayer is kind of moving and flowing. And those proteins can move within the bilayer. It's not like they're statically stuck where they are. So fluidity is simply the fluid motion, the movement of the membrane and the components within it. So then the question is, well, what affects membrane fluidity? Again, this is review because we talked about what makes lipids fluid versus more viscous or solid, as I mentioned, at room temperature. If you have more kinks in the fatty acid tails due to double bonds, those create unsaturated hydrocarbons and they do not stack well. That will increase membrane fluidity. Whereas if you have more tails with saturated hydrocarbons, you can imagine they're going to stack up nice. As I mentioned, those van der Waals forces or those not the van der Waals as much as the dipole moments are going to cause those tails to have temporary attraction to each other just due to random distribution of electrons at any point. And that's going to cause it to be more solid and held together, or as it's termed here, viscous. So the fluid mosaic model of membrane structure, which is what we're kind of discussing in general, is that the membrane flows and it moves and it has lots of components like a mosaic, right? A mosaic is, if you've done that in elementary school or even in high school, a mosaic is where you put a bunch of random parts together, right, that then come and give you an overall structure or picture. So it's called that because you have proteins embedded. You have glycoproteins and glycolipids and cholesterol molecules, which I'll get to in a second, and phospholipids, and all of these different types of molecules that are all kind of embedded in the membrane. And because of the nature of the phospholipids, everything's moving around and flowing. And remember, what dictates that flow is whether or not the tails are kinked or if they are more straight. And depending on the mixture of that, you'll get a more fluid membrane or a more solid or viscous membrane. So another thing that influences that membrane fluidity is a cholesterol molecule. Now, what type of macromolecule is cholesterol? Let's see if you can remember. Cholesterol is a lipid, right? The 4 fused rings, and you can see we have mostly carbons and hydrogens here. The only place that we have an oxygen is right at this location, and that draws a little bit of electron density. But what's interesting is you can actually see that how the cholesterol embeds is that OH group orients itself towards where the heads are because that's more hydrophilic versus the rest of the molecule, which is just carbon and hydrogen, so it's much more hydrophobic. What cholesterol does is interesting. It's a little different than what the tails do. So if you add cholesterol, it is a stabilizer of membrane fluidity. So at high temperatures, when you would expect molecules moving faster and you'd expect more sloshing or moving of the lipids, cholesterol will actually cause more dipole moments and cause it to hold it together, and it will actually make it less fluid at high temperatures. But at cold temperatures, it forces the tails further apart, and as a result, it flows better at cold temperatures. So it kind of has a stabilizing effect. But cholesterol is super important for your membranes. So yes, having high cholesterol is bad. Your cells make enough cholesterol for itself. It can synthesize it because high cholesterol will clog arteries. But cholesterol is absolutely vital for your membrane functioning and also for hormone creation as well. There are also membrane proteins. There are integral and peripheral proteins. Now, integrals are embedded in the membrane. I mentioned before that the core of it will have more hydrophobic amino acids in it, more hydrophobic R groups, whereas those peripheral proteins are going to be much more hydrophilic. Though the inside of the protein may be hydrophobic, you're going to have the ones that are associated with the side. If they're sitting in the watering environment, the outside R groups are going to be hydrophilic. They also sometimes will attach to lipids directly, the proteins. Sometimes they will attach to other proteins as well. It just depends. But if they expand the entire membrane, they're going to be considered integral. Integrate goes all the way through. If they do not span the entire membrane and just on one side or the other and attached, they're considered peripheral. Or like think about your peripheral vision, which means more towards the outside. So membrane proteins actually have lots of different jobs, similar to what proteins would pretty much do within the cell in general, right? They're involved in transport. That's a membrane-specific thing. They act as channels that allow certain molecules in and out, and they are very specific to what they let in or out. So different proteins will act as different types of channels for different types of molecules. There are also enzymes in the membrane. Enzymes drive chemical reactions that occur on the membrane for a reason. You have signal receptors. They receive signals so that when a signal binds to it, it will transmit that signal into the cell so that then there can be communication within the cell and there can be some sort of response associated with that signal that is sent. It's involved in cell-to-cell recognition, where some proteins will recognize the antigens on the surface of other cells, and we have self and non-self antigens. If it's a self antigen, the cell will be like, oh yeah, I should be near you. You belong to this area, right? And then there's non-self antigens on other types of cells that cells can recognize as being foreign. So so the proteins are involved in recognizing that, and that's a very similar relationship to the receptor example I gave you before. That this will signal a pathway inside the cell that allows it to know that it should be around those other types of cells, and sometimes even perhaps creating proteins to join those two cells together if it's a tissue. And this would be an example of those proteins. Proteins are used to stick the cells together. Like if you pull your skin, right, your cell doesn't just come off. There's an attraction there, and they're held together, and the cells are joined together through these proteins that hold cells together. And lastly, attachment to the cytoskeleton and extracellular matrix. If you're in my anatomy class, you may know what that is, but the extracellular matrix is the non-living stuff our cells secrete that acts as like buildings that our cells can live in, essentially. So like collagen in our nose is mostly not cells, it's mostly non-living matrix made of, or our cartilage, I should say, made of collagen. So the cells sometimes will attach to that as well, not directly to other cells. So those are just examples of a bunch of different functions that proteins have within the cell. Now we have to talk about the permeability part, the selectively permeable part of the membrane. So first off, easy things. What can cross easily through the membrane? Those would be the small hydrophobic molecules. Molecules that are small and hydrophobic that you have to remember: carbon dioxide is small and hydrophobic, oxygen gas is small and hydrophobic, and steroids are small and hydrophobic. CO2 and oxygen are super important because they're those vital gases that we take in if it's oxygen, and we breathe out CO2 as a waste product. But plants, obviously, they'll do cellular respiration too, so they will do that, but they also take in CO2 and create food from it. So the free flow of those molecules and the diffusion of those molecules easily across the membrane is very important. So small hydrophobic molecules, lipophilic substances, will cross easily. The heads don't end up being the barrier here because the heads will move apart every so often in the lipid bilayer, and as a result, lipids can sneak through. But the tails are going to shut out anything that is too hydrophilic. So hydrophilic things will not cross very well. And that now is dependent on the size of the molecule. So small hydrophilic molecules like water don't cross well. They'll sneak across occasionally, but not well enough to be physiologically relevant in terms of the processes that our cells need to do. So they usually need help getting across. We'll talk about that in a minute. So things that don't cross well would be like water, glucose, which is even worse because it's a larger molecule than water is and it's polar. Sucrose is even bigger, so that'll cross even more poorly. So large, small hydrophilic molecules that are polar do not cross well. As they get larger, they basically can't cross at all if they get too big. Things that cannot cross are things that are charged. Charged ions do not cross the membrane without help. They need a protein channel to get them across. So it's a continuum of difficulty crossing, with hydrophobic small molecules crossing easily. Hydrophobic large molecules can cross, but not a little bit worse than hydrophobic small ones. Small hydrophilic molecules do not cross well, but can cross occasionally. Large hydrophilic molecules don't really cross very well at all, and then ions do not cross. And so the two former examples, the small hydrophilic, a large hydrophilic, and ions usually need a protein to allow them to cross. So you can see examples of those ions: sodium ions, hydrogen ions, chloride ions, all do not cross without help. So the way I can remember this is large and in charge and polar do not cross well. Small hydrophobic things do cross well. Hydrophilic, difficulty. Hydrophobic, easily. So those large molecules, even those small polar molecules like water, they're going to need these special channels, as you can see here, to help get them across physiologically. The next thing we have to understand about the movement of molecules is what a concentration gradient is. And this is showing you what I mean by a concentration gradient. It's a difference between two locations in the concentration of a molecule or ion. So you can see on this side over here, we have a lower concentration of molecules, and on this side over here towards the right, we have a higher concentration of molecules. And what will happen is, due to just the second law of thermodynamics, as things spread out, molecules are going to move and even themselves out if a container is closed, right? So if I let this sit for a long time, and these are all gas molecules, or let's say it's a liquid, over time those molecules are eventually going to spread out as they bounce and collide due to kinetic energy that they have, and eventually they will even themselves out. But even if we have a membrane here, and let's say it's permeable to these ions, meaning the molecules will move from a high concentration to low concentration. It's just naturally kind of what happens. And you can kind of see that's what's happening in this GIF below here, is that the molecules are spreading out as they collide. So molecules will always go from a high concentration to a low concentration based on the second law of thermodynamics. So we're going to watch this video really quickly on YouTube. It kind of shows you.
And we're back. The team is looking down the barrel of a 4:55 meeting.
In this video, we will be discussing the structure of the cell membrane. When scientists looked at the selectively permeable cell membrane, they described its structure as a fluid mosaic. You might know that a mosaic is a picture made up of little tiles. The cell membrane is made up of different parts as well.
Bilayer. They're not having those molecules move very much.
The cell membrane has two layers of phospholipids, referred to as a lipid bilayer.
You can see the hydrophilic heads
The lipid
and the
bilayer,
tails.
the round head portions are hydrophilic, which means they are cell, are mostly made up of water.
Outside the cell environment, the inside the cytoplasm.
So the hydrophilic phospholipid heads of the outer layer will be oriented toward in a direction as far away.
You can see some of the types of things that embed, carbohydrates.
So, in the fluid mosaic boundary around the cell, however, other molecules are too big to fit through the cell membrane this way.
And then it's going to show you that.
So, how do these larger molecules pass through the cell membrane?
Or whatever, they have these
the molecules move through proteins
those
embedded in the cell membrane. Up of these proteins. We'll explore how things.
So let's now talk about that process. How do we get things across easily? So there's three types of transport. There's simple diffusion, which we talked about earlier, which is simply things crossing the membrane easily. These are our small hydrophobics. We have facilitated diffusion, which is the movement of larger molecules, but it's still going from high concentration to low concentration. It's still going across the membrane high to low, but it just needs help with a protein channel to get across. And lastly is active transport, which is where we're trying to go low to high against the concentration gradient, but energy is required to do this because we're kind of going against the second law of thermodynamics. We're trying to concentrate molecules from a lower concentration to a high concentration that always is going to require an input of free energy. So here's showing you that simple diffusion, small nonpolar molecules crossing easily until they reach equilibrium, and eventually they'll kind of stay at that equilibrium. Again, no assistance is needed for that. And the things that you want to remember, small nonpolar things, oxygen, carbon dioxide, steroids, those simply diffuse. You want to remember those three things. Facilitated diffusion, on the other hand, is crossing using help. Molecules that are large, charged, polar, as I mentioned, they need help crossing the membrane from high to low. There's both carrier proteins, which act as shut, like things that open and shut. That's a little bit more controlled. And then there's a channel protein, which just provides this larger opening and hydrophilic area for a specific molecule to cross. To go from high to low concentration, we do not need ATP, okay? High to low, no energy or ATP is required. We just need a channel because due to the properties of the lipid bilayer, these molecules cannot cross by themselves. Osmosis, which we're going to talk about more in depthly because we're going to actually quantify it. So it's a separate presentation and lecture. But osmosis is a special type of facilitated diffusion. Water is a small polar molecule. Can it leak across occasionally? Yes. Is it great? No. So it uses a special facilitated diffusion protein called an aquaporin. Aquaporins simply provide this hydrophilic zone for the water molecules to flow through easily. Without this, organisms would not be able to physiologically get water in or out very effectively. So water kind of simply diffuses across, or it appears that way, but it's because they have these special channels that allow water to cross. They do not simply go across the membrane. Remember, so osmosis is a type of facilitated diffusion. So if you mess with an aquaporin, if you mutate the aquaporin, you're going to impact the ability for water to move from a high concentration to a low concentration of water. But again, we'll talk about that more when we get into osmosis. Active transport, that requires ATP. You can remember that, active transport, ATP. This is when we're moving things against the concentration gradient. As you can see here, an ATP molecule comes in, we hydrolyze it, and it pumps the molecules across, concentrating them on one side of the cell or the other. So if a cell wants to get rid of wastes and there's more waste on the outside, they would do this. Or if they want to bring something that has a low concentration outside, but we need it in the cell in a high concentration, you would do an active transport mechanism. Again, remember, this happens because if we're going from low to high, it's going against the second law of thermodynamics. So as a result, we need to provide energy to do that process. It's not just going to happen naturally. So one of the classic examples that AP likes to talk about is the sodium-potassium pump. In my anatomy class, we talk about this through the scope of how our nerves reset, or our neurons reset when they fire. But the sodium-potassium pump is a kind of, it goes in both directions and pumps things against the concentration gradient. And what it does is it helps to maintain a charge on either side of the membrane, a positive charge on the outside and a negative charge on the inside. And that's important for, as I said, nerve firing, other cellular processes as well. So it does maintain that charge. But what it does is it takes 3 sodium ions and pumps them out of the cell, and 2 potassium ions and pumps it into the cell. So it essentially concentrates sodium on the outside against the concentration gradient and concentrates potassium on the inside, creating a concentration gradient there as well, or against the concentration gradient as well. It uses ATP for that process. As you can see at the bottom, you can see the ATP coming in and hydrolyzing it. It changes shape. It does a conformational change, pumps the sodium out, and then the potassium comes out in the other direction. Now this one's a little bit tougher. Co-transport. So co-transport is a type of, as I call, indirect active transport. So you're going to pump ions out of the cell using, so you could be protons, it could be hydrogen, so it's just another term for a proton, because that's all a hydrogen ion is one singular proton. It could be sodium ions, it could be any type of ion really. Usually we see sodium or hydrogen ions in this process. The purpose of this is to keep the concentration of the ion low inside of the cell, whether that be hydrogen, sodium, whatever. So you're always going to have a concentration gradient of things flowing into the cell. Now in this particular example, we see hydrogen ions flowing out, and then they're going to come right back in with the concentration gradient. So that should happen naturally, right? We don't need energy to do that once we used energy to pump it out, but it's going to bring something else with it. It's going to bring a sugar molecule, something larger, something that can't cross easily, usually against the concentration gradient. So you can see it's a little bit more, it's kind of like active transport, and this part is an active transport mechanism, but it has this second, more complicated step. This is called a symporter. It's a symporter because they're going in a similar direction. The hydrogen's flowing in and bringing sucrose in with it. Sometimes hydrogen will flow in and pump something out in the opposite direction. That would be called an antiport. But in both cases, they're using this potential energy of hydrogens flowing across and then the kinetic energy of them flowing across to bring something else either into the cell or get something out of the cell against the concentration gradient, something that isn't easily crossed. Okay. So as I said, the whole goal is to keep the concentration of the ion low here. Sometimes it's not just pumping it back out into the same place they're going to flow back in. Sometimes it's flowing out on another cell in the other direction. Like if you're going from the intestines, like hydrogen ions or sodium ions will be a high concentration inside the intestines, for example. Sodium will be, because you eat sodium all the time. Well, then we pump sodium into the bloodstream, not back out into the intestine, and as a result, it always keeps the low concentration of sodium in the cell, so sodium can flow easily from the intestine into your intestinal cell and bring other molecules that can't cross easily with it that we're trying to get in, like glucose, for example. Here's going to show you a quick video showing symports and antiports. What's nice is this one actually stays here. I don't have to open it separately. But you can see, so the sodium is flowing in and bringing sugar with it. So the sodium's a higher concentration on the outside and a lower concentration on the inside. And as you can see here, the sugar and the sodium are flowing in together. But then what they're going to talk about eventually, fast forward it a little bit here. You can see it keeps the sodium concentration high using that pump and, on the outside and low on the inside because we're constantly pumping the sodium out. Remember, the key is not necessarily that it's high on the outside. It does have to stay high on the outside, but we do need to constantly get rid of the sodium on the inside that is flowing in. Think of it like a ship that's gaining water or a boat that's gaining water. If it gains too much water, the boat will just sink. But if we want this boat to remain afloat or this process to continue, we have to keep bailing water out constantly, and that's kind of what's happening with this, this pump here. Now this is showing you antiports now. So you can see sodium's going in and calcium ions in this case are going out. Again, it's the only difference is they're going in opposite directions. This is still considered a co-transport mechanism. And remember, co-transport has the two components, the part where we're pumping something out using energy, and then the part where we are bringing something back in not using energy, but we're usually bringing something else in against the concentration gradient through that mechanism. So that's how we get individual, like small amounts of molecules across the membrane. Okay, but if we want to get mass quantities of things out using like vesicles and stuff, like pumping proteins that we've made out and things like that, we're going to use what's called the endomembrane system. And this involves really 2-3 organelles that are important, the nucleus as well. So once a message leaves the nucleus that a protein needs to be made, it goes to the rough endoplasmic reticulum and the ribosomes on them, or the ribosome initially isn't on there, then goes to the rough endoplasmic reticulum. The ribosome makes the protein, it spits it into the rough ER, the rough ER packages it and organizes it, ships it off in a vesicle, and then goes to the Golgi apparatus where it gets reorganized, repackaged, and then it ends up out of the cell. So we're going to talk about this whole process real quick so you can imagine it happening. So let's say there's an instruction to make a protein that leaves the nucleus. So that's called mRNA, if you remember from freshman bio or whenever you took it, if you took it in 8th grade. The mRNA will go to a ribosome. The ribosome will read it and start making a protein. If it is destined to be exported, like this one is, like a hormone, for example, or an enzyme in the digestive tract that we have to release it from the cells and it goes somewhere outside of the cell, then it will spit the protein into the rough ER where it's packaged up. Think of the rough ER almost like an Amazon warehouse, right? It gets instructions and then a bunch of proteins get made and it's shipped off. Well, then it goes via a vesicle from the Amazon factory to the, now obviously Amazon now has its own trucks, but let's just pretend. It goes to the Golgi apparatus, which is like the postal service, right? And that's what used to send Amazon packages. So the postal service will then repackage all the proteins that need to go together to one place, like into one mail truck, right? Not just the ones that were your order. And it'll then ship them together in one vesicle to eventually be exported from the cell. Or it could go into the cell membrane, or it just depends on the purpose of those proteins. So you can see here, it goes from the rough ER to a vesicle, to the Golgi, to a vesicle, and then it leaves. You always want to know that order. That's important to remember the order because it's always going to follow that order. Sometimes on the AP exam, they won't mention the vesicles, right? But it always goes from the rough ER to the vesicle, to the Golgi, to the vesicle, and out. Okay, or the rough ER to the Golgi and out if you disregard the vesicles. The mRNA itself, which is not protein yet, it's just the message for making protein, will start in the nucleus, it'll leave the nucleus, then go to the ribosome, and the ribosome will attach to the rough ER. Then the protein gets spit into the rough ER, vesicle, Golgi, vesicle, out. Okay, so you need to know that. I will definitely assess that on the test. But this is part of what's called the endomembrane system. Endo meaning internal membranes. And all of these, if we zoomed in, whether it be the nucleus, the rough ER, the smooth ER, the vesicles, have phospholipid, a double phospholipid membrane in them as part of them. So here's going to show you just a little bit so you can imagine this process a little bit better.
To study about the Golgi.
Turn the audio off here. So you can see the Golgi. You can see vesicles kind of moving here. Let's see. I'm going to try to find the part that I want you to see here. Okay, there's the plasma membrane, there's the nucleus, the rough ER. And you can tell it's rough because of the ribosomes on it, like that's why it's called rough. You also have the smooth ER as well. And you can see things will leave, those are the vesicles leaving, and then they enter the Golgi. Let's fast forward. Don't worry about the words here. It's beyond the course. In college, you'll have to know them. Come on. So here, let's see here. Come on. So there's the vesicle that's eventually going to make its way to the cell membrane, and then it's going to release the contents out. So you can see there's the vesicle going from the endoplasmic reticulum. It goes to the Golgi. The Golgi then shoots the vesicle out, and eventually it's going to, some of them are intracellular, some are extracellular, and it releases it. I don't love the way they made that vesicle look, though, because inside the vesicle it'll just release contents. It's not like the whole vesicle leaves the cell. Sorry, let's just go back into full screen mode here.
Okay.
Another term for the process of getting rid of mass quantities of stuff like this is called exocytosis. Exo, exit, cytosis, related to the cell, exiting the cell. And the way this happens, so those vesicles actually are moving along and they're being pulled by these special motor proteins called dyne and kinesin molecules. And they actually walk along using ATP. You can see it in that bottom left corner there, that every time an ATP is used, its leg, as I call it, will move and walk along the cytoskeleton. And it will pull vesicles to their appropriate spots, and that's their job. So again, this is all part of the endomembrane system. This is called exocytosis, and it requires energy to do this because we have to be able to move those vesicles around using those dyne and kinesin molecules. Endocytosis, on the other hand, is bringing, endo means in, bringing things in mass quantities into the cell. There's three main types. There's phagocytosis, there's pinocytosis, and there's receptor-mediated endocytosis. In all of these cases, again, they're kind of similar, but phagocytosis is where the cell is going to send out projections, grab molecules, and bring them in. Pinocytosis is usually when it's going to pinch in this way, okay? It's going to pinch in and kind of suck molecules into it as opposed to going out and getting them. To pine for something means to thirst, okay? That's why pinocytosis is cellular drinking. And then receptor-mediated endocytosis is similar, but in this case, molecules or ligands have to bind to receptors. And once that receptor binding occurs, the pit, which has these like little, it's called the coated pit. Again, they're not going to ask that in the AP exam, but that's how they can tell these types. They're specialized vesicles. But that only then will it pinch in and bring in molecules. So phagocytosis, cellular eating, goes up and grabs it, sends out pseudopodium, which means feet, okay? Grabs it, brings it in. Pinocytosis pinches in this way. And then, as I said, receptor-mediated, as you can see here, ligands have to bind to the receptors before it brings the things in that it's trying to bring in. And this is going to show you some actual phagocytosis. This is a white blood cell, and these are, I think, there's either streptococcus or staphylococcus bacteria. And as you can see, it goes out and grabs them and consumes them and breaks it down. You can see it happening here again. Goes out and grabs it and then breaks it down. One more time, you can watch it. It's going to go and get this one first. It sends out. It's like walking towards it almost. Right? See it? It's going to engulf it. Brings it in, breaks it down. That's phagocytosis. All right. So that is the lecture on cell membranes, its structure, its parts, how it works, its fluidity, the proteins embedded in it, what they do, as well as the different types of transport that you have to know for the AP biology exam.